All right, my clock just turned 12:01 East Coast time, 9:01 on the West Coast. So let's go ahead and get started. Hi, everyone. Welcome to today's CryoEM Service Centers webinar, organized by the 3 national centers established by the NIH Common Fund Transformative High Resolution Cryo-Electron Microscopy Program. So those centers are the Pacific Northwest Center for CryoEM, PNCC, located at the Oregon Health and Science University and Pacific Northwest National Laboratory in Portland, Oregon. And they have organized today's seminar. Also, the Stanford SLAC CryoEM Center, S2C2, located at the SLAC National Accelerator Laboratory at Stanford University in Menlo Park, California, and the National Center for CryoEM Access and Training, NCCAT, located at the New York Structural Biology Center in New York City. My name is Christina Zamani. I'm a member of the team at NCCAT, and I work primarily on the training side of the center, and I'm your moderator for today's webinar. This is our second installment of a monthly series. If you joined us last month, welcome back, and thanks for joining us again. If you missed last month's talk, we are recording and posting these seminars. So on the slide that's up now, you can see that link. We will also be sending this link in a follow-up email to everyone who registered for today's talk, along with links for our upcoming talks. So we have a regularly scheduled date, the last Thursday of the month, with the exception of November and December, which we've combined into one date, the first week of December. And so you'll get links to register for those in a follow-up email. We'll also be announcing those on Twitter and post to the 3DEM and CCP mailing list. So watch out for those announcements. Every month, we're hosting a user of the centers to present their work with a particular focus on cryo-EM methods. So along with highlighting the data that's coming from our microscopes, we hope these sessions are a useful resource for those of you who are newer to electron microscopy. And for that reason, we've made sure to leave plenty of time for Q&A at the end of today's talk. Before we introduce today's speaker, we'd like to introduce you to our centers so that you better know who we are and what resources are available to you. So with us today are Jeanette Myers, a scientific point of contact at PNCC, Corey Hexel, the operations director at S2C2, and Ed Ang, the manager at NCCAT. So they're each going to give a brief introduction from their centers, and then Jeanette will properly introduce today's speaker, Farzad Jalali-Yazdi, who has generously volunteered his time today to present his work that has made use of center resources. And finally, a couple of last logistics. Lauren Halesbeck, the project coordinator at PNCC, is here with us, and she is moderating our Q&A. You won't be able to turn on your microphones to ask questions, so we will be reading typed questions from the Q&A window. So please don't use the chat to send in your questions. Use that Q&A button at the bottom of your Zoom screen. Feel free to send in questions at any point. If our panelists can answer them for you in real time, they will in that Q&A box, and questions for Farzad will be saved for after his talk. You should also see the option to upvote questions that are in that box. So if you have a similar question or think a question is particularly good, upvote it, and that will help us prioritize questions for the end of the talk. So with that, I'm going to turn over to our center representatives, and Jeanette, take it away.
I'm actually going to be covering PNCC.
Oh, sorry about that. Lauren.
Okay. No problem. So hello, everyone. My name is Lauren Hilsbeck, and I'm the project coordinator for Pacific Northwest CryoEM Center, PNCC. So we're just going to go over a few things that we offer. So we do have 2 proposal types. We have a limited access proposal for preliminary data sets up to 24 hours, and a general access proposal for larger projects up to 480 hours a year for 2 years. Moving on to our microscopes, PNCC has one Arctica with K3 and 4 Cryos. 2 of those Cryos have Falcon 3 and K3, and another 2 Cryos have Falcon 3 and BioQuantum K3. We're planning on outfitting one Cryos with Falcon 4, fringe-free, and BioContinuum. And all of our Cryos will be updated with cold FEG coming the end of 2021. For sample preparation, we currently have a VitrBot and a Leica GP2 with a VitroJet coming in early 2021. We're really excited about that. How do you apply for access at our center? So at PNCC, applications are due every month on the 1st at 11:59 p.m. Pacific time. We currently leverage the EMSL user portal for all of our applications and proposal tracking. In general, it takes about 40 days from the 1st for proposals to be reviewed and receive an award letter. Once approved, there's a recommended user onboarding with myself. PNCC offers both screening and data collection of single particle analysis and tomography on approved proposals. Additionally, each approved proposal will be delegated a SPOC, a scientific point of contact, whom they can ask scientific questions. PNCC is currently under restrictions and is offering one-on-one remote training and small remote workshops. Once we can have visitors on site again, we plan to offer small on-site workshops covering microscope operation and sample preparation. Think about applying today. I'll introduce Corey for the next person speaking from Slack.
Hello, so my name is Corey. I work here at S2C2, which is the Stanford Slack CryoEM Center. Currently, we have 2 Titan Cryoses, one equipped with a K3 BioQuantum, and the other equipped with a Falcon 4. We are currently in the process of installing our 3rd microscope, which will be another K3 BioQuantum microscope. Currently, the Falcon 4 microscope has fringe-free, but all 3 will have it relatively shortly. We are currently taking applications for data collection as well as in-residence training. Much like the other centers, we currently aren't allowed to have people on site. So our in-residence training will be remotely initially and then moving back to on-site training once that's allowed. We also offer workshops. Our next one is middle of next month, middle of November, and details can be found online, and that's for a model building data processing workshop hosted by CCPEM. So if you have any questions, please visit our website and feel free to drop us an email. All right.
And up next is Ed.
Hey, my name is Ed. I am here at New York, where NCCAT is housed at the New York Structural Biology Center. We offer access to 3, soon to be 4 Titan Cryos instruments, as well as blot-free vitrification through a chameleon device. Something that we are really trying to promote is cross-training. And as this is COVID-19 times, we're under remote operations. But we have a full gamut of remote offerings. And if you want to learn how to use CryoEM in your research program, let us know. Apply now at nccat.nyspc.org.
Thank you all. So Jeanette, if you would like to introduce our speaker.
Yeah, sure thing. Yeah, so I'm Jeanette Spock at PNCC, and since I'm the Spock for Zad's project at the center, I'm going to go ahead and do his introduction. Hold on. Okay, so Zad received his BS degrees in chemical engineering and biology from MIT, where he worked with professors Langer and Seema to develop implantable devices to monitor biomarkers in cancer patients. He received his PhD from USC, working to develop and characterize peptide ligands using mRNA display. His research in Professor Roberts' laboratory focused on developing a peptide-targeted peptide ligand capable of stopping HCV virus particle production in human liver cells, as well as using mRNA display and next-gen sequencing to characterize the binding kinetics for thousands of ligands simultaneously. He joined the GROW lab in the fall of 2016 to study the structural mechanisms of neurotransmitter receptor functions by cryo-electron microscopy. His first publication focused on the structural mechanisms behind the zinc and pH inhibition of GLUN1/GLUN2A and DMA receptors. And he is currently working on understanding the mechanism of receptor activation. Okay, so take it away.
Can everybody hear me? Yes, good, okay.
Yes, we can.
Excellent. So thanks, Jeanette. As she said, I'm now a postdoc here at Oregon Health and Science University in the GUO lab, and I work on trying to understand NMDA receptors by cryo-electron microscopy. And today I'm really going to focus on how we were able to improve the resolution of our reconstructions from around 6 to 8 angstrom to much higher by the end of the talk. Okay. Let's see if I can go, there we go. All right, so first I'm going to talk about the first project that Jeanette mentioned, which is trying to understand zinc and proton modulation of NMDA receptors. And that's going to be kind of the starting point for the rest of the talk and in terms of improving the results that we got from that first project. Then I'm going to talk about symmetry and pseudosymmetry and when we think it's appropriate to apply symmetry to improve your resolution. Then I'm going to talk about optimizing our collection conditions and particle cleanup approaches and sample and basically skipping 2D classification, going straight to 3D classification. And finally, I'm going to talk about stabilizing the part of the sample that we were most interested to see and using nanodisk for that final boost in resolution. Okay, so this is what NMDA receptors look like. These are tetrameric receptors. They're found in the central nervous system. And alongside AMPA receptors, they're responsible for most of the excitatory neurotransmission in the central nervous system. NMDA receptors are obligate heteromers, meaning they're often composed of 2 GLUN1 subunits and 2 GLUN2 subunits. Here, GLUN1 subunits are shown in blue and GLUN2 subunits are shown in orange. I try to keep the same color scheme for most of this talk. And I also have like a little cartoon of the receptor down here. So when I talk about a specific part of the receptor, I'll just point to this cartoon. So these are receptors that are embedded in the lipid bilayer in the membrane, and they stick out of the membrane. So there is an extracellular portion of the receptor, and then there is a transmembrane portion of the receptor. So the transmembrane part is called the transmembrane domain. And there's also a cytosolic part of the receptor, but these tend to be poorly structured. And for most of the structural studies, these are cut out. The extracellular domain of the receptor has 2 subdomains. First, the amino-terminal domain, and these are more distal from the membrane, so they're farther away from the membrane. And this is where a lot of allosteric modulators bind and really help guide the function of the receptor. This is very opposite of AMPA receptors, where the amino-terminal domains are still somewhat of a mystery because they don't really seem to modulate the function of the receptor. And then going a little bit closer to the membrane, you have the ligand binding domains. And this is where the natural ligands for the receptor, which are glycine and glutamate, bind. And binding of these ligands leads to opening of the ion channel gate in the transmembrane domain and allowing sodium and calcium to go through to the cell. So the first structures of these receptors were solved in 2014, simultaneously pretty much by the GUO lab and Furukawa lab. And both labs used the GLUN1, GLUN2B subtype of the receptor for their crystallography experiments. Now, in order for their crystals to go to high resolution, they had to introduce a series of mutations, including this K216C mutation at the amino-terminal domain. So this is the receptor if you're looking at it from the top, just looking at the amino-terminal domain. And what this mutation did is it locks the amino-terminal domains of GLUN2B together at the central axis of the receptor. And this really allowed both groups to go to relatively high resolution of 3.7 angstrom and be able to build the model for the crystal structure. Okay. But what the GUO lab also noticed is that when they removed this mutation, they saw 2 different conformations in the asymmetric unit. And I think that's fascinating. We'll talk about it later. Okay. So this is where we stand. Unfortunately, all of these mutations, including this K216 mutation, made the receptor inactive. So you were no longer able to apply ligands to it and obtain current. So the gate seemed to no longer function. So fast forward to 2 years. So this is 2016. Both labs again came out pretty much simultaneously with 2 different structures, this time focusing on the allosteric modulation of these receptors, the GluN1, GluN2B receptor. And this time both labs moved to cryo-electron microscopy. And the first thing you notice is that the resolution of these receptors is now substantially worse. So these were generally at around 6 to 8 angstrom resolution. But the reason that the labs moved to cryo-electron microscopy is that they were able to reverse all those mutations that rendered the receptor non-functional. So now you have fully functional constructs, and they were able to show how allosteric modulation happens with this receptor. Okay, so this is also where I kind of joined the lab, and my first project was trying to understand the molecular mechanism for zinc and pH sensitivity of the GluN1, GluN2A subtype of the receptor. And by electrophysiology, it was long known that the GluN1, GluN2A subtype of the receptor can get inhibited by zinc. So when you apply agonists, glycine and glutamate, to this receptor, you get this inward current. However, even with single-digit nanomolar concentrations of zinc, you start inhibiting this current. And there are 2 really interesting things about this inhibition. First, the extent of inhibition is not complete. So even at saturating zinc concentration, you still end up with a significant amount of residual current. And the second interesting thing is that this residual current is a very strong function of pH. So at high pH values, the residual current is pretty large. So the extent of zinc inhibition is pretty small. And at lower pH values, the residual current is pretty small, so the extent of zinc inhibition is fairly large. And we were trying to understand how this happens at a molecular level. So what we did was we expressed the receptor in hex cells, we solubilized them in detergent and purified it, and our final cryo-EM sample was in 0.1% digitonin. And we made grids at around 3 to 5 milligrams per mil, which was pretty typical for these type of receptors. For our collection condition, we were more focused on trying to gather as many particles as we could. So we went to a relatively low magnification. So we were collecting at 1.7 angstrom per pixel on a K2 sensor in super resolution mode. Okay. And in terms of data process, I would say our data process pipeline was pretty typical. It probably is pretty typical for today for most groups, which is we take the movies that we collect, we motion correct them, we estimate the CTF, then we template pick, or we use a Gaussian picker or a template-free picker to pick particles. Then all of these particles will be 2D classified to get rid of junk particles. I usually did 1 or 2 rounds of 2D classification. And then these particles are fed to an ab initio model generator, or a similar receptor structure is filtered to remove all high resolution details. And that gives us our initial 3D template. And that initial 3D template and the cleanup particles together will be used in a refinement to give us a 3D reconstruction and refined model. We then focused on the extracellular domain of the receptor because we were trying to get away from all the noise that was in the detergent micelle that we obtained with these receptors. So we focused on the extracellular domain, and we did extensive 3D classification to further clean up the data, get rid of junk particles, and also parse out any potential heterogeneity or multiple conformations if we had that in our sample. And then particles that belong to classes that showed similar conformation were combined, and we finally obtained our final reconstructions. Okay, so the first structure that we obtained was for the receptor in the absence of zinc at pH 7.4. The receptor looks pretty typical for NMDA receptors. We still have the amino terminal domain here on top. We have the ligand binding domain here in the middle, and the transmembrane domain that was embedded in the detergent micelle. And we also noticed the conformation of the amino terminal domain. So the isolated amino terminal domain crystal structure was already solved by the Furukawa lab, and they had structures in both presence and absence of zinc. And the amino terminal of GluN2A receptor has these 2 subdomains. So there is one domain here, another domain here, and they form this clamshell-like structure. Zinc binds at the cleft of this clamshell, so you can think of this as Pac-Man eating those little dots. And when zinc binds, it leads to the closure of this clamshell. And we noticed that in the absence of zinc in this structure that we had, the clamshells were open and we didn't see any density that would be correlated to zinc. Okay, and then we added 1 micromolar zinc to our sample and we got a second set of structures at pH 7.4. And this time after 3D classification, we noticed that we were able to isolate 3 unique conformations. So what do these look like? Well, in the absence of zinc, so we had EDTA to take away all zinc, we noticed that with the glue N2A, as I said, the clamshells seem to be in the open conformation, and there is no zinc at the cleft of the clamshells. To me, we also noticed that there are 2 alpha helices at the central axis of the receptor, where these 2 subunits are interacting with each other. And to me, this really looked like 2 fists kind of coming together, doing a fist bump with 2 knuckles touching each other. So we call this the 2-knuckle symmetric or 2-knuckle sim state, because both sides are unoccupied by zinc. And then we looked at the 3 structures we got in the presence of 1 micromolar zinc. And the first structure, we noticed that there seems to be zinc bound to 1 clamshell because the distance between these 2 sublobes is now smaller. But the other one, the distance between these 2 lobes was still pretty large. So it looks consistent with 1 lobe being in the zinc-free state and 1 lobe being in the zinc-bound state. We still saw those 2 alpha helices interacting at the center of the receptor, so it still looked like a 2-knuckle conformation. So we call this the 2-knuckle asymmetric because in terms of zinc binding, they looked asymmetric. The next conformation, we noticed that now both lobes seem to be in this closed and zinc-bound conformation, and there's a rotation that allows only 1 alpha helix at the center of the receptor to interact. So we call this the 1-knuckle. So it looks like a fist bump with only 1 knuckle touching the other knuckle. And finally, in the last conformation, these subunits move away from each other. There is no interaction at the center, and we call this the extended conformation. Okay, so throughout this project, we collected 10 different data sets at 3 different pH values, multiple zinc concentrations. And that really gave us an ensemble of structures. So we got 18 structures belonging to 7 structural classes. And when we put it all together, we noticed that there is basically a set of structures that represent the not inhibited or the less inhibited structures. And there are a set of classes that represent the more inhibited or the very inhibited structure conformations. And what zinc and pH do is that they shift the equilibrium of the receptor if you go to high zinc or low pH more towards these ensemble of structures that are more inhibited. And under a lot of conditions, the receptor can actually sample many different conformations, and they exist in an equilibrium. And all adding zinc or lowering the pH does is shifts the equilibrium so the receptor can more easily sample those structures. So we were pretty happy with this set of results. We published it. But if anybody knows Eric, they know that he always likes to push for high resolution data. And I think we had a long way to go. So we were starting the highest resolution structure that I had in this project was around 5.1 angstrom. And I'm showing it here. It belonged to this 2-knuckle sim, so not bound by zinc conformation. And you can see, some helical pitches. The helical pitches aren't great, and some density for maybe bulky side chains. But again, there is a lot wanting in this structure. But the biggest issue for me was that for the next part of my project, for the next project in a way, I was really trying to focus on what's happening at the transmembrane domain. And for that, this was where the weakest density for the structure was, as well as the lowest resolution. So the TMs, some of the TMs in some of our structures just refused to show up. Some of these TM helices, and the resolution overall was generally around 6 to 6.5 angstrom or worse. Okay, so we knew that we wanted to do something about this. So the first thing that we tried to do about this was to see if we could apply symmetry. Now, the receptor has molecular symmetry. The 2 gluon 1 subunits and 2 gluon 2 subunits are identical molecularly. And overall, it looks also like it has structural symmetry. Now, the structural symmetry that it looks like it has is C2 symmetry. And to figure out if this was something that we could do, I forced C2 symmetry in one of our reconstructions. So this is just a test data set. So when I force C2 symmetry, I noticed that I go from about 6.6 angstrom resolution to 6.1 angstrom. So that's a half angstrom improvement in resolution by forcing symmetry. So that's pretty good. But even more importantly, we noticed that the density features, especially at the transmembrane domain, are much stronger. And we're able to see things much better now that we've applied C2 symmetry. So we get an increase in resolution, we get a better density, but are we actually allowed to do this? Is this something that is valid to do? Well, what C2 symmetry means is if when you take your receptor and you rotate it 180 degrees along this axis of symmetry, you get the same structure back. And when we look at this C2 forced structure, that's what we see. So when we rotate it 180 degrees, you get the same structure. Now, this is cheating because we force C2 symmetry. So by definition, this has to do that. So what happens if we do this to the structure we got when we didn't apply symmetry? Well, so let's rotate it around the central axis. And now you can see that it doesn't look identical. So especially at the amino terminal domain, you see that the receptor is doing this funny little dance where one GluN2 subunit seems to be higher than the other GluN2 subunit with respect to its LBD. So now we were wondering, okay, is this an artifact that we are seeing of data analysis? Does the receptor actually have symmetry, but due to some heterogeneity being in our particles, we are seeing this, is false pseudo-symmetric state? Or is this something real? Is this something that's a function of the receptor? Okay, so we had one tool in our toolbox, and that was because out of the 10 different data sets that I had analyzed in this first project, 6 of them contained this structure, this 2-knuckle sim conformation that we were most interested in pursuing. And because we had 6 of them, we could now kind of try to parse out whether this difference, this deviation from asymmetry is something that we see consistently. And if it is, then maybe it's a function of just how the structure of the receptor is. Or if there's a lot of noise and maybe it's just we are at the limit of our resolution or data analysis capabilities. So when we measured the angle that the ligand binding domain and the amino terminal domains make on the 2 different subunits, on the 2 different GluN2A subunits, we noticed that this angle is extremely consistent. It is, so the difference between those was very statistically significant. So it really looks like this is a function of the receptor itself. So if you apply C2 symmetry and improve your resolution and maybe even improve your density, the structure that you're getting is not a real structure. It's this weird Franken structure that you've constructed, and you can't actually look at a cell and find that structure. Okay, so symmetry is out. How else can we improve things? This next thing that we looked at was our data analysis pipeline. So this is a pretty typical approach that, as I said, so we used to do in our first project, and I think a lot of other people still do. And I have a test data set here to demonstrate my point. So I start with 280,000 particles. I use a Gaussian picking algorithm to pick particles, and I do 2D classification into 200 classes. And as anybody who's done 2D classification knows, you end up with some classes that look like your receptor or your protein, and some classes that really just look like junk. So during this first round of 2D classification, I always try to be as non-stringent as I can. I try to take as many classes that even have a hint of maybe some of my receptor particles are in there. So if you look at some of these, selected classes that I have here, you kind of have to, strain your imagination to see if there are any good receptor particles in there. But this is generally what I do. I try to take as many classes as I can in that first round. Then I follow that by a second round of 2D classification. And in this case, I'm usually a little bit more stringent. And I try to pick classes that really look like they have my protein in them. And even now, you can see some of these classes might be a little questionable. So then I take these cleaned up particles, which at the end I ended up with 65,000 particles. And I do ab initio, and I generated a 3D model that kind of looks like my structure. So I was pretty happy with that. And then I combined this data sets with that 3D model, and I refined the structure to 6.4 angstrom. So this wasn't bad. This is somewhat typical for what we saw. But I then decided to see, all right, what happens if I get rid of the second round of 2D classification and replace it with a round of 3D classification? So when I did that, I ended up with one class that had good particles. I actually ended up with less particles. So the first 2 rounds of 2D classification gave me about 64,000 particles or 65,000 particles. Now I have 56,000 particles. So I ended up with less particles, but this map refined to a much higher resolution of 5.1. So I went from 6.4 to 5.1. The map looks better. The density features on the map look better. So that was a pretty big improvement. So I thought, all right, so if I go from 2 rounds of 2D to 1 round, then I get an improvement. what happens if I go to no rounds of 2D? So then I went ahead and did straight 3D classification with all of these junk particles that were picked with particle picking. I just straight went into 3D classification. I ended up with a single class that showed good features, had 77,000 particles. And when I refined it, I'm now at 4.5 angstrom. So again, this is a pretty huge improvement over the last analysis pipeline, where by eliminating this 2D classification, I end up with a higher resolution structure. And finally, I did a second round of 3D classification, got rid of another 11,000 particles. The resolution didn't improve, but I think the density features improved slightly. All right, so let's take a look at these two classes. So on the left, I have my initial, my data set that was initially cleaned up with 2D classification, followed by a round of 3D classification. I ended up with 56,000 particles at 5.1 angstrom resolution. And if you look at the orientation of the particles that make up this structure, you notice that it is kind of patchy. There are certain orientations that seem to be missing. But on the other side, I have my data set that was cleaned up initially by 3D classification, followed by a second round of 3D classification. I ended up with 66,000 particles at 4.5 angstrom. And if you look at the particle orientations, it is much more even. So the particles are, now you're getting a lot more even sampling of the different orientations you had in your data set. Now, these two shared around 44,000 particles. So this means that this one had 12,000 unique particles. This one had 22,000 unique particles. And when we trace the origin of where these particles were lost, out of those 22,000 unique particles, 21,000 particles were lost in that initial round of 2D classification. And that's even so, I tried to pick as many classes as looked like they might even have my receptor in there. So yeah, so 95% of the good particles were lost in 2D classification. Now, what we noticed is that there are other people in the field who are noticing this as well. So I really just love the title of this paper that came out about a month ago by the Donev group on bioRxiv. And the title is Routine Sub 2.5 Angstrom CryoEM Structure Determination of this Family of GPCRs. And if you look at their method section, they say all 3 data sets were cleaned solely by 3D classification. So, and to be honest, if you think about it, it also makes a lot of sense, because what 2D classification, what the ultimate goal and the incentive for 2D classification is to find similar particles, average them out, increase the signal to noise, and at the end give you good looking 2D class averages. And what 3D classification's incentive is to try to move particles around and find the right orientation for the particle until it can give you the best 3D class that it can. And if you think about the ultimate goal of cryo-EM, is usually not to get you very good 2D classes, right? The ultimate goal of cryo-EM is to get good looking 3D reconstructions. And for that, really the incentive falls along line 3D classification. So I think we've tested this on multiple data sets with NMDA receptors. We've also tested it in the lab with a lot of different proteins. And this really seems like the way to go. We haven't found a single case where 3D classification gave us worse results. Okay, so moving forward, what else can we do? Well, we can always try to add more particles. And in this case, we also tried higher magnification. So fortunately for me, when I started the second project, it was also when PNCC was upgrading their, at the time, only Cryos, but now they have 4, from a K2 camera to a K3 camera. And there are several advantages of a K3 camera. One, it's about 4 times faster. So you can go to higher doses without having to worry about coincident electrons. And that means that you can actually capture your images much quicker. The second thing is that the sensor itself is about 60% larger. So you get more image per image. So that means per image, you're covering a larger area. And they also claim it has a higher DQE. I don't really have a way to test that. Okay. So for our first data sets, because we were at relatively low magnification, what we had to do was we would move the stage, we would focus, then we would take one relatively low magnification image. And that image was taken on a K2, and it covered basically most of the hole because we were using the 1.2 micron holes. But for the second project, we started moving to the 2 micron holes, so either 2.1 or 2.2 holy carbon grids. And we also went to higher resolution, higher magnification. And what this allowed us to do was take multiple images per hole. So we call this multi-shot. And later we also started image shifting into multiple holes and taking multiple images. So now we have this multi-shot, multi-hole strategy where we would move this stage, we would focus, then we would take 45 images, then we would move this stage again and focus. Now, this made screening a little bit of a difficulty because I still use this approach, but for even screening different squares, and it just became time consuming. But the data collection rate is blazingly fast. So we went from collecting around 600 images per day at 1.7 angstrom per pixel to around 8,000 images a day at 0.8 angstrom per pixel. And because this was at a relatively low magnification, we were getting up to 80 particles per image. But for these higher magnification image, we are getting about 25 particles per image on average. And these represent particles that have survived 2 rounds of 3D classification. So these are generally the particles that end up in our final reconstruction. And when we put it all together, we went from collecting around 50,000 particles at low magnification to collecting 200,000 particles at higher magnification. And that means that each particle, we believe, brings in more useful information. Now, can we quantitate that? The answer is kind of yes. So if we look at the Rosenthal-Henderson plot, where you plot the second root of resolution as a function of natural log of particles, the slope of this plot can give you the B factor. And for the first set of data that we collected at low magnification, the B factor was 350. And that means that you need to add an astronomical number of particles to be able to reach 5 angstrom and 4 angstrom and above. But for the second set of data, for the second project, the different data sets that we collected, we noticed that we'd be able to improve the B factor to around 120. So not only are we adding more particles, each particle is also bringing in more useful information and contributing more to our final reconstruction. So we went from having a ceiling of around 5 angstrom to having a ceiling of around 4 angstrom. What does the map look like? Much better. So the maps are now much improved. The helical pitches are pretty deep. You can get very good side chain density. But there is still something wrong. And what's wrong is that the transmembrane domain still has pretty weak density. And also the linkers that link the ligand binding domain to the transmembrane domain are pretty, consistently not showing up in our structure. So it looks like we have an extracellular domain that's just floating in space and a transmembrane domain. Now, if you contour this a lot lower, you see those linkers, but overall, we weren't very happy with this. So we knew we had to stabilize this part of the receptor, this transmembrane domain. And we were fortunate enough to have an in vitro assay that we could kind of assess what was going on at the transmembrane domain. And we were able to find a compound that allowed us to do that and actually stabilize the TMD. Now, this was the only data set that we collected at NCCAT, thanks to Ed and Alina. And all the other data sets that I'm showing were captured at PNCC, and that's really convenient because they're just down the hill from us. But once we started analyzing the data, we were really blown away by how much sharper the density was for the transmembrane domain. So we were seeing all the helices, the density for them were showing up in a continuous way. We were seeing linker densities that link the LBD to the TMD, so this ligand binding domain to the transmembrane domain. Again, something that we hadn't seen at these resolutions. We were seeing linkers that link the various transmembrane domain helices together. Again, something I'd never been able to resolve for the NMDA receptors. So this compound really helped us stabilize the transmembrane domain and allowed us to really image this part very well. This was a relatively small data set, and it went to 4.7 angstrom. It was collected on a K2, but at higher magnification. So we were pretty happy with the results that we got. But the question is, all right, we still want to push this further. We want to get better data, higher quality data, higher resolution. So what do? We had kind of agreed that one of the problems we seem to be having is that our detergent purified sample, even when we had the concentration at relatively high, 3 to 5 mg per mil, we still, when we went to look at it under the microscope, we had to go to relatively thick ice to be able to get good particle distribution. And the particles didn't seem to be able to distribute and appear in thinner ice. And we thought that that might be hurting us in terms of our, reaching the resolution limit that we want to reach. So the question is, how do we get the receptor to go into thinner ice? We tried different approaches, different grids, different additives, and nothing really was giving us, helping us out. But we decided, okay, so let's move away from detergent and let's see what that does. So we started reconstituting the receptor into these lipid nanodiscs, and this is now done pretty routinely in our lab. And what we do is we basically take the detergent purified protein, we take the nanodisc protein and solubilized lipids, we combine them all together, then we remove the detergent by biobeads. And this allows the nanodisc protein to form these lipid nanodiscs. And our receptor also can get incorporated into that. So you no longer need detergents to keep the transmembrane domain stable. Finally, you purify the complex by size exclusion chromatography. So we did that. We took this peak, we put that on a grid, and it looked atrocious. It looked really, really bad. Didn't matter if we go to thin ice to thick ice, you basically get a carpet of particles. You can't see any side views. Maybe we are looking at a lot of top-down views. Maybe we are looking at a lot of aggregated protein at the water-air interface. We can't tell. We just know that this isn't it. This isn't going to give us a high resolution structure. And it doesn't matter even if we go to lower protein concentration. So then we said, all right, so let's put in back a little bit of detergent. So we put in FOM, which is a fluorinated octyl maltoside. And these fluorine groups stop this detergent from intercalating into the lipid bilayer or into my lipid nanodisc. So it's able to help break that air-water interface a little bit, or the aggregation at the air-water interface. And now we are able to see individual receptor particles. We are able to see side views. So we were pretty happy about this. We also noticed that the more of FOM we added, the more particles went away. So we went with like the Goldilocks amount of 100 micromolar FOM. We collected an overnight data set. This time, so most of the analysis I was showing you with the 3D classification we had done in CryoSpark. This time we didn't rely on exact same map or exact same pipeline. It doesn't matter if you use Relion or CryoSpark. You can just skip 2D classification, go straight to 3D classification. Usually with Relion, you manually have to add more iterations. So after about 100 iterations, our classes had converged in terms of the fraction of the particles. And when we look at the class that had particles in it with the features of our receptor, we further 3D classified it. There was one class that showed really strong linker densities. We took that and we refined it. And this is the final map that we have in hand. And this really blew us away. So this map, the overall structure is at 3.1 angstrom, and this is a fully functioning functional construct. And I'd like to remind you that the crystal structure for this receptor is at 3.7 angstroms. So you can see that the core of the receptor is at substantially higher resolution than 3.1. And even at the transmembrane domain, even the outer helices are at worst at around 3.5 angstrom. So if you focus on the transmembrane domain, you notice that you can see side chain densities, you can see continuous helices. It looks great. You can even find these cholesterol-looking molecules that are adhering to the outside of the transmembrane domain. So we were really, really excited about this. So, and what I ended up doing is I actually had to de novo build from scratch most of this model, because we noticed that the rigid body fitting that we had done for the transmembrane domain of our low resolution constructs were pretty far off from the mark. So now, again, you can see even on the outer helices, you can find density really well, and you can fit this density, you can fit your model into this density pretty well. Okay, so there were still, so we wanted to look at how much better this data set was from all our other data sets. So going back to the Rosenthal-Henderson plot, you see that this stands head and shoulders above even our higher magnification data set that we had collected before. So not, we are at a reasonable number of particles. Again, this was from a single overnight data set, data collection session. And so even though we are not at a ridiculous number of particles, we are way above where all our other data sets have been in terms of the resolution. So we were pretty happy about this. But there were still two questions I wanted answered. The first question is, are we changing the conformation of the receptor by putting it in a lipid nanodisc? So are the structures identical or not? And that kind of would go to answer my question of, are the previous structures we had obtained in detergent valid, or are they somehow distorted? And when we overlaid the digitonin structure with the nanodisc structure, we noticed that they are identical. So all the helices are located exactly the same place for both of these structures. So the only difference really seemed to be the resolution. So that going to the nanodisk allowed us to go to a higher resolution. And it's really made me happy because that means that, all of those structures that we had obtained in detergent also seem to be valid. And then the second question is, I really want to put a nail in the coffin of symmetry of this receptor. So I took the same particles that I had resolved without symmetry, and I forced them to refine with symmetry. And overall, the resolution doesn't change very much. It's about 3.1 for both structures. So we are not getting a boost in resolution. But if you look at the density, you notice that without applying symmetry, the density is much better. Especially looking at the amino terminal domain. On this side, you can see individual helices and side chain densities. And on this side, it really is just a smear. And it's not just at the amino terminal domain. At the ligand binding domain, you also see the smearing here, whereas here, it's pretty clear. And finally, even at the transmembrane domain, there isn't much smearing. But when you fit a model to this, you notice that the helices have moved when you force C2 symmetry. So again, to me, this was the ultimate confirmation that this is really not a C2 symmetric structure. This structure has deviation from symmetry. I think it's fascinating. I don't know why it has deviations from symmetry, but it is clear that it does. And this also kind of points back to that when the Guo lab had tried to crystallize the NMDA receptor and had removed that disulfide bond at the amino terminal domain, they saw two different conformations in the asymmetric unit. So I think even from the earliest data sets in crystallography, we can kind of find out that maybe this receptor isn't actually symmetric. Okay, so what helped us go to higher resolution? Using higher magnification, increasing our data collection rate, skipping 2D classification and cleanup, going straight to 3D classification, stabilizing the transmembrane domain, the product of the receptor that we really wanted to see using a specific compound, using nanodiscs for better particle distribution and potentially giving it a native lipid-like environment, adding FOM so we could actually visualize the nanodisk samples under the microscope, and only applying symmetry if it's valid to do so. And for us, that means if it improves your density and resolution, that's probably not a good enough basis to apply symmetry. And what you really need to do is you really need to refine the structure without symmetry and then measure the deviation from symmetry. And only if that deviation is very small are you then allowed to apply symmetry. Okay. So I'd really like to thank Eric for his patience, a lot of patience and guidance throughout all these projects and allowing me the opportunity to work on these exciting projects. I'd like to thank the Go Lab members, past members of the Go Lab, some of whom I saw in attendance today, PNCC staff and NCCAT staff, as well as my funding sources and resource sources, which are NIH, HHMI, PNCC, and NCCAT. All right, and thank you for attending. And I'd like to answer any questions you might have.
Hi, Zad. So one of the first questions we received was why stage position data collection? Why not use image beam shift to collect more data?
So that's exactly what we ended up doing. So in the initial set of experiments, we would, let me see if I can go back up there. There we go. So in the initial set of experiments where we were just, moving the stage, collecting data, it was very slow. And then later on, we were moving the stage, focusing, and then using image shift and beam shift to collect up to 45 images. And that really sped up our data collection. So that might have been an earlier question that was answered later on.
So the second question we got was, what are the lax 2D classification parameters that you used?
So, I mean, this is pretty based on each individual's judgment call. But for me, by lax, I meant out of the 200 classes, I picked particles in classes that looked remotely like they had my receptor. So if there are programs where you can give a 3D class, I think CryoSpark can even do this now. You can give it a 3D class and it slices the receptor and tells you what the different projections of the protein or the receptor would look like. So if you look at your 2D classes and you say, all right, so what is that? That clearly is not my receptor. It's too small, the density is too hard. I can't imagine any orientation of my receptor that would give me that projection. So I'm gonna exclude it. But at some of these classes like this one, I was like, all right, like I can't really identify my receptor, but I can potentially see, all right, maybe there's a micelle here, maybe there's ECD here, but I'll give it the benefit of the doubt. So I was very, very non-stringent. And I wasn't collecting just particles that look like my receptor. I was trying to collect anything that might even remotely be my receptor. That's what I mean by lax.
The next question we had was, what does the shape of the FCS or FSC mean for the quality of the new higher resolution particle?
This might help if I knew which which one they're talking about, but let's, sorry.
I think it was the question was asked around one of these slides.
One of these slides. Okay, so when the FSC improves, that means that you have a higher correlation at that value of resolution. So by going from these lower correlation, and you can see that the FSC isn't, I mean, ideally FSC would be very, very high around 1, and then it would jump straight down at the limit of your resolution. But it usually has a slope, and when the bottom part of it is extended, you start looking at it and saying, all right, maybe that's not quite real. There are FSC wars that are going on the email lists. Feel free to attend them. They're fantastic.
A little bit more clarification for this question. They said they're referring to the linear looking curves.
Linear looking curve. I'm not sure what they're referring to.
5.1 resolution.
5.1 resolution. Okay, here. Oh, okay. So kind of at the edge here. So again, this is what I was saying, that when you get this flattening, especially around the 0.143 cutoff, that's when you kind of say, all right, maybe this isn't really at 5.1, or maybe it's at 5.1, but you start questioning things when you get this sort of flattening. But when we went to this high resolution, you can kind of see that it crosses the threshold at a pretty steep angle later on. This is pretty typical in terms of the FSCs that I get. If I go to the final map here, again, you kind of have this this flattening here at around 8.5 to 4.2, but it's still at really high correlation, and then it drops pretty sharply. So it's something we've seen. We don't know if it's due to the micelle and any noise that the micelle generates, but it's pretty typical for most of the receptors that we're dealing with.
All right, the next question was, do you think this would work well, skipping 2D with samples with less recognizable features that are harder to align?
So we've done this on some test data sets with smaller samples that have more of them inside the micelle. And if you start with a reasonable initial model, I think it's actually more helpful for those samples than these bigger samples. Because with these bigger samples, you would think that the 2D classes should be easier to distinguish. But if all you have, for transmembrane domain, for transmembrane proteins that have a transmembrane domain, is, if most of it is buried in the detergent micelle, it seems like 3D classification would be the better way to go, because a lot of 2D classification might not be able to give you the features very easily. So I think you might have to test it out and see what you see. Especially if you're using CryoSpark, they've made this relatively computationally cheap, because I can classify 1 or 2 million particles into 8 different classes in less than a day on a single GPU, which is pretty fantastic.
Great. And then the next question is, when tracking back the unique 22K particles that were lost during 2D classification, did you run 2D on those unique 22K and see classes today looked unique? Or did they look like something that you would throw away?
So that's a good question. I haven't done that. That's a good point. The problem is, once you're below 50 or 60,000 particles, it becomes harder to get good looking 2D class averages. So I can go back and actually do the 2D classification and see what they look like. But all I know is that even when I was trying to be as non-stringent in my selection, so selecting classes that look like this, or even look like this, these particles didn't make it into my selection.
All right, the next question we have is, did you ever do 2D classification of the poor 3D classes to see if you still had good particles not going into the best 3D classes?
I have done this in the past, not specifically in this test data set, but I've done that a lot, especially because sometimes you get two classes, not just one. And like, let's say one of them is at 6 angstrom, one of them is at 8 angstrom. So then I do generally like a combination of things. I refine each class individually, then I combine the two classes. I've also done 2D classification of the worst class to see if there are some particles in that worst looking class that I can combine. And I can tell you that in almost every single case, anything that you combine with your good class, especially if the difference between two classes is relatively large, of like 2 angstrom or so, anything that you add to your good class reduces your resolution. So, and nowadays I do this on a lot of different data sets, because some of my data sets I like to break up into smaller pieces and analyze individually. So each of those I then go and like do this sort of analysis for, and almost always, and even if you look at the 2D classes for a class that looks similar to your receptor, you end up seeing like 2D classes that look like your receptor. So then you would think that if you combine it, it might be better. So the final resolution might be better. But in every single case, when I combine them, it gets worse.
The next question is, where was the binding?
Lauren, I'm
Yes.
just going to interrupt you for 1 second. We're coming up on 1 o'clock. So for anyone who has to head out for their next meeting, I just wanted to say thank you for joining us. Zad has said he can stay a little longer to answer more questions. But for those of you who can't stick around, we'll keep the recording going and hopefully get to a few more questions. Expect a follow-up email with links to the recording and registration for upcoming talks. And thank you for joining us today. And I will turn it right back over to questions.
Great. So the next question was, where was the binding site of the compound that helped to resolve the TMD and the linker between TMD and LBD?
For that, you have to wait for the publication to come out. This is like 80% unpublished data. So I got the permission from Eric to talk about it, given I wasn't gonna reveal some of the details.
The next question is, if class 2D is skipped, how to get the initial 3D model?
Okay, so that's a really good question. And even for myself, one thing that I still do is I do go through this pipeline. So I do 2D classification, I like 1 or 2 rounds of 2D classification, and I get my ab initio. But then at this point, what I do is I ignore all of the 2D classification I've done, and I go to the initial set of particles and I do 3D classification. So even today, I still do 2D classification to get my ab initio, but then I ignore all the results from 2D classification and I just go straight from the initial set of particles and I do 3D classification. You can also just get, if the protein is something you worked on before, you can always just start doing a filtering of another structure and use that as your initial model.
The next question was, the reason for selecting FOM, did you go for detergent screening?
FOM is something that has been tested on multiple data sets, multiple publications. So this was kind of our first thing that we tried. We also know that even sometimes when people have preferred orientation problems in detergent or proteins that are not in detergent, but they have preferred orientation problems, FOM is usually the #1 thing that people test. And what we really wanted to do is we wanted to make sure if we are going to use a detergent, it wasn't going to destabilize our lipid nanodiscs. Because that's the worry that you have with detergents, right? That if they start solubilizing your lipids away from these nanodiscs, then the whole point of using a nanodisc kind of goes out the window. So we had to find something that acted like a detergent, but wasn't able to intercalate into our nanodisc. And we also have the radioactive binding data showing that adding FOM doesn't actually destabilize the receptor. So we had that data beforehand when we started doing the screening.
The next question was, excellent work. For the 3.1 resolution map, did you see the zinc density? Why state move, not beam shift move?
Okay, so I think why stage move and not beam shift move was also answered above, which is for these higher collection rates, we have to do beam shift and image shift. So that is actually what we did to increase our data collection rate. But in terms of zinc, no, because these were obtained in the absence of zinc. I'm more interested in receptor activation, and that happens in the absence of zinc. So we were going away from that. But we did see density for the ligands at the ligand binding domain. So all 4 ligands we were seeing density for, so the glutamate and glycines.
The next question was, did you stabilize the transmembrane with the compound with the nanodiscs?
Yes, that's a good question. And yes, this was with the compound and in the nanodisc.
I had another question. Do you use initial model generation from your own EM data for the 3D classifications? And then can you use model derived from other known crystal or EM structure, pros and cons of each approach?
So that's a good point, that's a good question. We generally try to get the model from our own EM data. In the past, I have used models from either previous data sets or from, actually even recently, I've used models from a previous data set as long as I know that they're basically the same protein under similar conditions. Because your initial model should be filtered to relatively large numbers in terms of resolution, so relatively low resolution, you shouldn't have to worry too much about how, as long as the overall shape of the receptor at like 10 or 12 angstrom is the same, it should be okay. So I think up here, I kind of have an idea. So this could be the initial model you start with. So you can see it's very blobby. There are very few details at maybe 10 or 11 angstrom in terms of resolution. So that's really all you need. Now, I'd be a little worried if you lack the detergent micelle. If you're trying to use something from a PDB, you can generate a model from a PDB. But even then, if the extracellular domain of your protein is pretty large, then even if you lack that detergent micelle, you might be okay.
Another question was, how efficient is the nanodisc incorporation? Say, are 80% of receptors successfully embedded?
Good question. I think what I found throughout doing this several different ways is that about 50% of receptors incorporate into the nanodisc. And the reason, and that includes all the losses that I gathered throughout this process, because when I normally do affinity purification, and initially when I was making grids, after affinity purification, I would do size exclusion chromatography, and I would take the peak of that and I would make grids. Now, when I'm doing this nanodisc reconstruction, I do affinity purification, then I do nanodisc reconstitution, then I do one size exclusion chromatography. So I don't do a size exclusion in between. And I noticed that initially when I would do this, just affinity purification followed by size exclusion chromatography, I would lose about half of my protein. But when I do affinity purification, nanodisc reconstitution, and size exclusion chromatography, I get back about 25%. So I lose 75% of the protein. So that means that the nanodisc reconstitution step and all the losses that you acquire and whatever else there is loses about 50% of my protein.
The next question is, it seems that ECD is highly flexible relative to TMD, and wonder if you tried multi-body refinement. Also wonder if you noticed any lipids from nanodisc.
So the second question is easier to answer. The answer is yes. We definitely see lipids. They look like cholesterol. They're not very smooth. They look kind of like flattened, and they look like they have this, like long noodle shaped form. Body refinement, that's a really good question. I spent a good amount of time on this. So this is actually have a slide on it too. So we try to define the extracellular domain and the transmembrane domain separately. And first I started with the whole receptor, and the whole receptor was about 3.3 angstrom in Relion. This was a slightly earlier version. And when we look at the whole receptor, but just looking at the resolution with this ECV map, it's better. So it's 3.23. And when we look at the resolution at the TMD level, it's about 3.8. So it's worse. So again, the good part and the bad part together gives you this medium number. So now we decided to see, all right, so what if we just refine the ECD? We get around the 3.2 angstrom. So that's an improvement from this 3.23 to 3.18. Not a huge improvement, but a slight improvement. But what happens if we refine just with the TMD, with C1 or C2 symmetry, and it got significantly worse? And then finally, we looked at multi-body refining and analyzing the ECD and the TMD separately at the end. And the ECD refinement basically matches the ECD resolution. The TMD refinement is even worse than the separate TMD refinement. So at least in our initial setup trying to do multi-body refinement, it really wasn't fruitful. So, and I think there's a lot of micelle dust. And I think the ECD can actually help align the stuff in the micelle better than when you do have this micelle stuff around and trying to refine only this region. But that's been my experience so far. Anytime I try to do refinement of transmembrane domain or a specific part of it, it usually blows up.
All right, another question was, what membrane scaffold protein did you use for the nanodisk?
What membrane scaffold? I think they mean like what nanodisk protein. Again, I'm sorry to say this, but this is one of those details that we're gonna put in the manuscript. I just want people to know that there are many options. There are saposins, there are peptidiscs, there are MSP proteins. There's even SMA, which questionable if you can call it a nanodisk. So you might have to try a few things to figure out what works for you. But if you want to know what worked for me, just unfortunately, you have to wait for the manuscript there.
Another question was, would you recommend skipping 2D classification for small soluble protein samples less than 200 KD?
I am gonna say, I don't know if I can't think it would hurt. So I would definitely try it and see what you see. You could play around with the parameters a little bit, number of classes you give, and the binning as well as the box size. But I would give it a try. I can't imagine it hurting. If 2D classification works, then 3D classification theoretically should work too.
Another question was, sorry. Does beam tilt refinement help to improve the resolution for a data set collected by beam shift?
So we collect our data with CryoEM. And CryoEM, you already do this sort of calibration to try to calibrate coma versus image shift, coma versus beam tilt. We've tried in Relion as well as in CryoSpark with the beam tilt refinement. I don't know. I feel like those are the things that help you get you from like 2.2 angstrom to like 1.8 angstrom in resolution. And so far, when we are kind of even at around 3.1 angstrom, we haven't seen an improvement from those. One thing that really helps us is per particle CTF estimation. And that's both CTF and astigmatism and everything. That usually gives us a pretty big boost. But particle polishing or beam tilt refinement at the resolutions we haven't seen an improvement. But also we do a pretty finer patch motion correction. So we used to do 5x5, then went to like 9x5. Now we are doing like 14x11. So these are pretty small squares. So that already looks a little bit like per particle motion correction to some extent. So we haven't noticed any improvement from those.
All right, Zad, let us know if you want to stop with questions. There's quite a few more.
Okay. I'm happy to answer maybe one or two more, and then anybody, I think you guys have my contact information. If you have any questions, please feel free to email me.
Another question was, I was wondering if you observed more drift on 2.2 grid compared to the 1.2 or 1.3.
Not noticeably, no.
In terms of pursuing higher resolution, how many particles are reasonable?
Very good question. And actually, the paper that I talked about here on bioRxiv, they actually go through and talk about how many particles they had. And I think there is a different paper on bioRxiv. If that person can email me, I can direct them to a paper. But that paper talks about individual elements. So one of them was using using an energy filter, whether that helps or hurts, using a volta-phase plate, whether that helps or hurts. And one of the measures that they had was how many particles before you get to 3 angstrom resolution. And for a lot of, and I think it was in GPCRs, but for a lot of those data sets, around 150 to 200,000 was where they were. Some of them were as low as like 120,000, and these are all for non-symmetric proteins. And for my own data set that went to 3.1 angstrom, it was about 150,000 particles that went to 3.1 angstrom. But that is after throwing away quite a few particles. So I usually end up with maybe 10% of the initially picked particles, maybe 15%. So after all of that, I ended up with 150,000 particles. All right.
Probably the last question we're going to do is impressive data as usual. Thanks. Any comment on when we could enjoy it at EMDB/PDB?
If anybody would like to put some pressure on Eric to get this out, they're welcome to try. We're working on it, but it is probably a little while out before you guys see it.
All right, I think that's going to wrap up our Q&A since we can't get to all of these.
I also noticed there were a couple of questions about the recording. We will post the recording on that same YouTube channel that I gave the link to before. We'll send that information out with a follow-up email as well. So you should be able to find it. Yeah, so thank you so much, Zad. That was amazing. I think if we were all in a room, you would be getting a rousing round of applause right now. Thank you very much.
Thank you very much.
Hi, this is Mike Schmid, and I have some information about next month's talk. If you want to give a preview of what that might be, is that okay?
Oh,
Christine?
yep. I'm still recording, so go ahead and let us know.
Great. Okay, next month's talk will be given by Sudha Chakrapani from Case Western. It'll be December 3rd, 9 a.m. So in a departure from a normal November and December time, we've combined them. It will be December 3rd, Thursday, at 9 a.m. Pacific time. And the title of her talk is Understanding Gating Mechanisms in Pentameric Ligand-Gated Ion Channels by CryoEM. And the data was collected here at the S2C2. I'm looking forward to sharing that with you. Thank you.
Thanks, Mike. We hope we see you all then.